students of honors chemistry. I thought I would video our first lab together to see how this goes. We've talked about this in class already today. Our first experiment is going to be a paper chromatography experiment, and hopefully you are ready to roll. So this is my demonstration for you. This is the book that we're using. It should have come with your microchemistry supplies, the kit that you ordered for this class. And if you open that up to page 16, you'll see the beginning of the experiment in terms of the list of materials and the procedure for us to follow. All right, so we're going to start off just by running quickly through our reagents and equipment list to make sure that you have everything ready. And as we go, I will point out a few tips that I want you to remember as you're doing this experiment on your own later. We'll start with the good stuff. We're going to be using 3 solutions that you need from your microchemistry kit. There's a lead nitrate solution, nice pretty light blue copper nitrate solution. These are our 2 active reagents. We are going to be mixing those together in a solution and then using our paper chromatograph to separate the components again. Potassium iodide is what we're going to use after to develop our paper chromatograph. So make sure you keep your solutions straight so you're not getting them mixed up. While we're mentioning the solutions, I'll just point out a couple of tools that you're going to use. The first is a 96 well plate. In your kit you would have received 2 plates. Make sure you use the 96 well because its little wells are small enough to mix the very small amounts that we're going to be using today. And to obtain our solutions out of their jars, we're going to be using what are called mini pipettes. And it's important that you are careful with these. Pipettes are very commonly used in chemistry labs. They come in many different sizes and versions depending on what you need to measure. With these pipettes, do give them a good rinse between uses with some distilled water. So I'm going to use 1 pipette to measure my solutions. I'm going to rinse well in between, and then I'm saving a second pipette to use with the potassium iodide. I've put a little mark on my second pipette. I just don't want to contaminate my reagents. It could mess up this lab, or it might cause problems for future labs if we're using the same jars of reagents again. So even though this is a basic experiment and you're doing it in your home, good technique is important, and you can develop those technique skills even in this setting. So good to pay attention to those details. The other reagents on our list, you can experiment. This is where you can add things that you think of. I've brought some blue food coloring. I brought other colors, but since we don't have a very big piece of paper, I'm only going to use the blue. And then a felt marker, which we can use as well. Over here, I've added a cotton swab that we're going to use so that that's out. And then sliding over here now to more of our equipment. This is the 150 ml beaker that your book, the manual, suggests you use. And basically what we're going to be doing is putting our reagents on the bottom of this paper, wrapping that up, stapling it together, and then dropping it inside this beaker for the distilled water to wick up the paper and move our reagents across the paper. So you can go ahead and do it that way. I'm going to also experiment with a slightly different setup where I'm going to be taping the paper. Actually, I'm going to use my more narrow paper. Taping a paper to a little dowel here. It's actually a skewer. You can use a barbecue skewer or a straw. And I'm just going to suspend it that way in the water, and that just allows me to have the paper out flat, and then I can see what's happening. Whereas when it's wrapped up, it'll be a little more difficult. Not a big deal, but I just thought I'd have some fun doing it that way. Other items that we'll need, some distilled water in my wash bottle. I'm going to use that to rinse out my gear before I use it to make sure I don't have anything that can contaminate the reagents, and some extra little beakers. And then of course we're going to need a ruler to take some measurements when we're done, pencil to mark some markings on our paper, and then some extra things like scissors and tape. You can also use a stapler to fasten your paper, whatever you find easier. Okay, So if you think you have everything ready to roll, we can go ahead into the procedure now. Okay, now that we have everything set up, we're going to get the ball rolling. The first thing we're going to do is set up our paper chromatographs. These are these square sheets of paper that you should find in your kit. And I'm going to do two. I've made one a little more narrow so I can hang it in my my wide mouth jar. And then this one we're going to leave as a square and set it up as the book instructs in our 150 ml beaker. I've already put a little bit of water at the bottom of each of these. You want to make sure that the water, the level is about 3 to 4 millimeters, and make sure that the water covers the entire bottom of the container. Some containers have a cupped bottom, and so you don't want to create a place in the center where there's no liquid. And the same thing with the jar. So now that our liquid's set up, I'm going to go ahead and set up our paper chromatograph. There's a diagram in your book on page 17 how to mark your paper, but it's pretty straightforward. We want to line 1 centimeter up from one of the edges. And we're using pencil because pencil is really graphite, which is a non-polar material, so we can be assured that the graphite is not going to get taken up in the water as the water diffuses up our paper. And please be sure that you're using distilled water. You don't want to have any impurities in your water that will affect the experiment. And we're going to make some markings on that line that's 1 centimeter from the bottom of the paper. I'm going to have three different samples, so I'm going to make 3 marks, and they're going to be about 2 centimeters apart. So I just picked about a 6 centimeter piece of this line in the center, and I'm marking markings 2 centimeters apart. I'm going to make 4 markings actually, because I want one that I'm going to consider a control, which will just be where my pencil mark is, in case for any reason my pencil manufacturer has lied to me and there's more than graphite in this pencil lead. And I'm also going to put a little tick at the top corner on the left-hand side of my paper. I've just put an L because once this gets wet, I'm not sure what's going to happen or how the paper will appear. So this little L at the very top will hopefully help me keep my bearings because the paper is going to get pretty wet. All right, so our paper is all set and ready to go. It's time to get our reagents out. So the first thing we're going to do is take our copper nitrate. And I mentioned using pipettes for getting the copper nitrate solution out, but I've discovered that the dropper in the bottles is really quite a nice fine dropper. So I think if you're careful, you can squeeze quite easily 3 drops into a well. Make sure you take note of what well you use. I'm going to drop into H12 because I'm right-handed, and it's just a lot easier to aim for that lower right-hand bottom. So I'm going to put 3 drops right into the center of the well. Very nice to control, so no need for a mini pipette for that. That was my copper nitrate. Now I'm going to double check that I'm grabbing the right clear solution, lead nitrate, and I'm going to put 3 drops in the same well, H12. All right, that's 3 drops. Now I will use a mini pipette to stir H12 up. And I've got a little bit of distilled water sitting in a beaker over here, which I'm going to actually rinse my mini pipette. I'm going to drop a little bit of distilled water and discard it over here. And that's just so that that mini pipette, I can use it again another time and not worry about contaminating my next experiment. And I've put the distilled water that I used to clean my mini pipette. I just discarded it in a completely separate container that I have no plan to draw any water out of, so I don't get contamination that way. So now that that is mixed up, we can start to apply it to our paper. I'm going to start with the solution. I'm going to use the same pipette that I just used to mix, and I'm going to draw up what's in that well. And then just apply 1 drop. All right, so I'm going to apply the drop of our lead nitrate and copper nitrate solution to my second dot. I'm only going to apply one drop because I want each drop to dry after I apply it. So that's going to be drop number 1. I'm going to let that sit for a minute and dry. While it's drying, I'm going to apply a spot from the dry erase marker, the felt tip marker. I'll just give that a noticeable spot there and let that dry for a second. And then finally I will do 1 drop of food coloring. I'm not sure how this one's going to come out, so I'm actually going to use a little pipette. And I just put 2 drops of the food coloring on there. We'll see if that's enough. And I'll just give it a minute for the first drop of my copper nitrate, lead nitrate solution to dry, and then I will apply a total of 5 drops, and then we'll set it in the water. Okay, so now everything's been applied to the paper. It's important to let it dry in between drops, and the reason is that if you apply 1 drop, especially of your copper nitrate, lead nitrate solutions, and the drop spreads out when you put it on the paper. So if you let it dry in between, you will minimize that spread. If you just keep adding your 5 drops one on top of the other, it would spread right across your paper and not make it very useful to read once it's done. So it's now that everything's been applied, I'm going to go ahead and curl it up so that it will fit into my beaker. I'm going to use tape because I can't get my stapler inside. And I do need it to be as flat as possible for the bottom edge, so you want to try to hold your cylinder nice and flat at the bottom so that it sits level in your distilled water. And then I just take that and drop it right into the beaker, and you can see the water start to diffuse up the paper. And I'm going to do the same thing with my second paper. Grab another piece of tape. I've already checked that the length of this paper is the right length to hang off of my dowel, so I'm just going to put a piece of tape on the dowel. Alright, so now that water is going to diffuse up this paper, and as it goes, the idea is that it's going to separate components of mixtures, and it's going to separate them based on physical properties. In this case, we're hoping that there are things that have different molecular mass, molecular size, so that the heavier items are going to move more slowly than the smaller items, and that's how we're going to carry out their separation using the paper chromatograph. So we're going to give that about half an hour, the book tells us. We'll wait and see how long it takes. The most important thing is that you make sure you pull it out of the water, pull your paper out of the water before the water diffuses right to the top. You want to leave a few millimeters at the top. And when you pull your paper out, you want to make sure you mark the level that the water diffused up to. You can draw a line with pencil across there, because we're going to need to take that into consideration when we look at our results when this is all done. Okay, so a few things have happened. The first is that the water has diffused up both of the paper chromatographs. This one I stopped when I had about 10 millimeters to go. This one, which was wrapped in the 150 ml plastic beaker, had about 5 millimeters to go. And I was glad I did mark the upper left-hand corners of these, because once they were wet, it was hard to tell which side was which. So now, what we can see right off the bat is that my felt marker separated into different colors. You can see each constituent color, that there are several pigments used to generate that black color, and you can see that the pigments have separated. My food coloring apparently looks to be quite pure. I've just got a big blue blob that migrated up the paper almost right to the top. So what is in this area is our copper nitrate, lead nitrate solution, and we can't see them, they're clear. So we're going to take our third reagent, the potassium iodide, put a few drops into a plastic beaker. I've already got my cotton swab sitting in there because in fact it's the cotton swab I want to moisten. So I'm just getting that nice and wet. And then I'm going to rub the potassium iodide on where I expect my copper nitrate, lead nitrate solution to be. So I've marked its spot here. And we'll add a little bit more. And I will do it on this one as well. All right, so now we've rubbed the potassium iodide onto our paper in the area where we expected the copper nitrate, lead nitrate solution to diffuse up the chromatograph. And it's important to take an observation before the potassium iodide dries. And essentially what potassium iodide does is it's going to cause the copper nitrate to turn brown, which you can see mine actually migrated off the paper. And the lead nitrate has turned yellow. On my other paper here, you can see the brown is at the top and the copper nitrate, and the lead nitrate is below, closer to my my line of origin, and it is yellow. So although we mix them together in a solution and put them in a well, we've now separated them on our paper chromatograph. The diffusion time was about 35 minutes, and then I did use a blow dryer to dry my papers to speed up the drying process so that I wouldn't have to wait for those papers to dry. And once the paper was dry, I was able to rub the potassium iodide to find my copper nitrate and lead nitrate. And you can see just as we're talking, the potassium iodide is drying and the copper nitrate is disappearing. I can no longer see the brown spot very well. So that's why it's important to get those observations in fairly quickly. And when you make those observations, do a marking around your copper nitrate and maybe do a circle where you have the most intense color. We're going to take a couple of measurements from those circles. The first measurement will be to the middle of the lead nitrate circle. So where we drew our yellow circle around the yellow, I'm going to measure back to the line of origin. I'm just going to take it to the nearest millimeter, 8.1 millimeters. And my copper nitrate, I've got 8.7 millimeters. And I'm just going to jot those down. Copper nitrate, lead nitrate, 8.1, 8.7, and my total diffusion distance of the water itself is 9 millimeters. Okay, so we'll head to our computers and just walk through making a couple of calculations to look at working with a paper chromatograph.